guys, so we're going to start with our review for the AP Bio exam with Unit 1. All right, covalent bonds, remember, are when electrons are shared between two atoms. Atoms in a molecule can attract electrons in different degrees. So this is determined by how electronegative an atom is. So the more electronegative it is, the more strongly it pulls those electrons to itself. Covalent bonds, those sharing of electrons, can be nonpolar, which means the electrons are shared equally, or it can be unequally shared, which then would create a polar covalent bond. So that would create this uneven sharing of electrons, which would make a partial positive portion of the molecule and then a partial negative. So here we have water. Right, the oxygen is more electronegative, so that has a slightly negative charge. The hydrogen is slightly positive. It doesn't get the electron as often. So polar covalent bonds contribute to this kind of arrangement here where the electrons are orbiting the oxygen more frequently, giving it that partial negative charge. This contributes to hydrogen bonding, which then gives water its unique properties. So hydrogen bonds form when hydrogen bonds to a more electronegative atom. Again, we just talked about that. Typically, we're going to talk about it with water, so we're talking about hydrogen and oxygen. Just remember, it can be between hydrogen and nitrogen as well. And these are not, right here, this is not a hydrogen bond, right? This is a polar covalent bond here. The hydrogen bond is between one water molecule and another separate water molecule. All right, we're going to come back to this. We're going to keep talking about how shape determines function or structure determines function. I know we've talked about that a lot, so I just wanted to keep that in there. All right, so that hydrogen bonding contributes to cohesion or water molecules sticking to other water molecules. I think of like co-captains, so same team. So water molecules are adhering to each other. That kind of leads to like the beating of water on cars. It also helps with the transport of water against gravity in plants. Adhesion, another property of water. This is when water clings to another substance. So this would be like when you put a paper towel in water and you see the water going up the paper towel, that's adhesion. And then we have surface tension as well. So how hard it is to break the surface of a liquid. So why those basilisks can run on the surface of water, right, is due to surface tension. All right, moving on to carbon compounds. So organic compounds have carbon in them. We have carbohydrates, lipids, proteins, and nucleic acids. So all of these we kind of talk about are biomolecules, right? So monomers, we're going to talk about the monomer unit of each one of those biomolecules, making it into that macromolecule or polymer. Polymer is just another name for those longer molecules. All right, so remember, we can go back and forth between monomers and polymers using those in different reactions. So a dehydration synthesis reaction is when two monomers are going to form a polymer, right? And that is through the loss of a water molecule. So you can think of dehydration, water loss. Polymers can be broken down by hydrolysis. So you can think of we need a water, hydro, to lyse or break the bond. So we're going from something bigger to something smaller here. Both of these, as well as any reaction in your body, are helped by enzymes which speed up the chemical reactions. We'll talk about enzymes more as we go, but it's important here as well. So here you see the dehydration reaction, two smaller molecules being linked into a larger polymer. This water comes out. So the water comes from a hydrogen and a hydroxyl group of these two molecules being joined. Hydrolysis, when we have a longer molecule here, we have to add water to break it, right? So hydrolysis, hydrolysis, we break it into smaller components. All right, the first of the biomolecules that we're going to talk about is a carbohydrate. So carbohydrates are sugars and starches. Carbohydrates are made up of carbon, hydrogen, and oxygen. So CHO, this is coming back to, I know this was a long time ago, but monomer unit of a carbohydrate is a monosaccharide. Typically, we just say glucose. So glucose, and then there is also fructose that is a monosaccharide as well. So you can see here's glucose and fructose. Again, these are coming together through dehydration synthesis, right? Water is coming out, and then we're forming sucrose, which is a disaccharide or two sugars. So glucose, fructose, and sucrose, monosaccharides and disaccharides, are typically their function is going to be immediate energy. There's also polysaccharides, which are many smaller subunits together. So these are mostly used for storage. So storage or long-term energy. Starch is polysaccharides in plants. Glycogen is storage in animals. All right, lipids. So lipids are also made up of carbon, hydrogen, and oxygen. So CHO again. Remember, lipids though tend to have less oxygen. So carbohydrates, when you look at them, you can see there's quite a lot of oxygen. So typically carbohydrates, we said, were the 1 to 2 to 1 ratio, 1 oxygen to every 2 carbons to every 1 hydrogen. Lipids, on the other hand, are mostly carbon and hydrogen, those hydrocarbon tails with just a little bit of oxygen. So we'll look at that in a minute. Lipids can be kind of subdivided into fats, phospholipids, and steroids. We're mostly going to talk about fats and phospholipids. So fats are made up of, their monomer unit is a glycerol and fatty acids. So glycerol and 3 fatty acids. And then when we look at it, so here is the glycerol, right here's glycerol, and then here is the fatty acid tail. So you can see again, see how many carbons and hydrogens there are and just a few oxygens in here. The difference between the different types of fats, so saturated fatty acids have no double bonds, and then unsaturated fatty acids do have a double bond. So you can see here again, here's the glycerol, here's the 3 fatty acids, no double bonds in there. Unsaturated fat, here's our glycerol, here's our 3 fatty acids, and you can see there is a double bond. Major function of fats is energy storage, long-term energy storage. All right, and then phospholipids. So phospholipids should give you an indication, right? It is a lipid. It also has a phosphate group on it. These are really important because they make up cell membranes. So remember the structure of a phospholipid. We have that hydrophilic head that faces out, and then the hydrophobic tail that faces in. And then the phospholipid bilayer, right? There's 2 layers that make up the cell membrane. Okay, so proteins, moving on to proteins. Proteins are really important, have a lot of different functions in the body, including defense, storage, transport, movement, chemical reactions, right? We already talked about that they are enzymes as well. So proteins, sometimes we call them polypeptides, sometimes we call them a chain of amino acids. So proteins, the monomer unit of a protein is an amino acid. Amino acids are made up of an amino group, carboxyl group, and then a side chain. So we now have CHO and then N, chome. Okay, so those are the elements that make up proteins. Remember, sometimes there can be other elements like sulfur in there, but typically CHON. So this R group can be many different things, and this R group is really what determines the way that the protein is going to fold and interact. So those R groups can be nonpolar or hydrophobic. They could be polar, which would make them hydrophilic, or they could be acidic or basic, which would give them a negative or a positive charge. So just remember that this determines the structure of the protein, and I'll get back to that in a minute. So amino acids are linked by peptide bonds, and you can see each polypeptide has the same sequence. Each polypeptide has a linear sequence of amino acids with a carboxyl end and an amino end, right? We talked about that already. So you can see here's a peptide bond. The new peptide bond is forming, again, by dehydration synthesis to make a larger polypeptide. And remember, even the peptide bonds are still covalent bonds. So anytime that there's a dehydration synthesis reaction, it's forming a covalent bond. All right, and protein structure determines its function. So there's different levels of protein structure. So we have the primary structure, secondary, tertiary, and then sometimes there can be a quaternary structure. That is just if there's more than one protein that join together to form the final structure. So in our primary structure, that is just those peptide bonds linking those amino acids together. It kind of looks like that beaded necklace there. Secondary structure is formed by those hydrogen bonds. So secondary structure could form this alpha helix or beta pleated sheet. Tertiary structure, then, that is the bending where it gets its final shape. Remember when you guys did that with all the tacks, right? And that is determined by the R groups, those functional groups, and how they are going to interact with each other. And then again, sometimes we can have that quaternary structure if we have two proteins joining together. All right, proteins can be denatured, which means they lose their structure, they kind of unravel, and that could make them inactive. That denaturation could be caused by really big changes in pH, salt concentration, temperature, or some other environmental factors. All right, last biomolecule that we're going to talk about are nucleic acids. These could be DNA or RNA. The monomer unit of both of these is a nucleotide, right? A nucleotide is going to be a nitrogenous base, a sugar, and a phosphate group for both of them. Differences between DNA and RNA, the sugar, right? Sugar in DNA is deoxyribose, and RNA, it's ribose. RNA is a single strand, DNA, double strand, right? Double helix, which then that double helix, remember, runs anti-parallel. We have those two strands kind of running opposite. The other difference between DNA and RNA are the bases, right? So if we have thymine in DNA, and then in RNA we have uracil. So the pyrimidines are the bases that are just a single ring, cytosine, thymine, uracil, and then the purines are these double rings, adenine and guanine, and then remember the base pairing rules. So A pairs with T in DNA, or A pairs with U when we're making mRNA, and then C pairs with G. All right, so that is Unit 1. I hope that helps you guys review. All right, bye.